है ना?
उसमें तो लिखा भी है ना 14 सितंबर करके।
हाँ 14 एट 12:30, 12:49 हो चुका है। अच्छा करने दीजिए, उनको हो जाएगा। वो
हाँ हाँ।
हो जाने के थोड़ी देर बाद से दिखना स्टार्ट करता है।
2 वेटिंग दिखा रहा है अब।
हाँ 2 वेटिंग।
व्हाट्सएप पे भी भेज दी हूँ मैम आपको, नहीं गया था अभी।
मुझे शेयर कर रही हैं क्या मैम आप?
हाँ मैं भेज दी हूँ मैम, मेल में भी और व्हाट्सएप में भी।
हाँ हाँ, आ गया आ गया।
आ गया?
यस
मेरा आ गया।
यूट्यूब में, हाँ यूट्यूब मेरा भी आ गया।
थैंक गॉड।
बोलिए मैम। गुड।
थैंक यू मैम।
थैंक यू मैम।
मैम को कॉल करूँ ज्वाइन करने के लिए?
हाँ कर दीजिए।
हाँ
ओके। फिर वो भी अपना मैम मीनवाइल आप पोस्टर
हाँ मैं शेयर करती हूँ मैम। पोस्टर विजिबल है?
यस मैम।
स्क्रीन कर दीजिए मैम,
क्या मैम?
फुल स्क्रीन कर दीजिए।
हेलो। हाँ अर्ची मैम ज्वाइन कीजिए ना।
सीपी मैम माइक।
हाँ यस मैम बोलिए।
माइक
माइक क्या हुआ?
बंद तो कीजिए। बंद तो
म्यूट कीजिए मैम,
कीजिए।
म्यूट कीजिए।
बंद कर।
शतविशा मैम,
जी मैम।
आप शेयर कर देंगे पोस्टर का, क्योंकि अगले दिन मैं ये करूँगी ना, तो
हाँ
फिर ज्वाइन टू परमिशन के लिए अगर फिर मैं वो नहीं कर पाऊँगी।
ओके,
पोस्टर स्क्रीन पे है ना?
ठीक है मैम। हाँ मैं कर देती हूँ।
हाँ मैं आपको शेयर करती हूँ।
सतीशा मैम आपके पास है या आप सेंड करूँ?
मैं सेंड कर दी मैम।
अच्छा ठीक है।
शतविशा मैम,
अर्ची मैम। हेलो। अर्ची मैम। Archie ma'am? Hello, Archie ma'am? Hello. Hello, Archie ma'am.
हाँ मैम।
Okay, good afternoon, ma'am. Okay,
गुड
within 2 minutes, we will going to start this.
ओके ओके।
Okay. Anupam ma'am, can we start now?
Yes, ma'am.
हाँ मैम.
Just wait for a minute.
Yes ma'am.
Just 1 minute.
Anupam ma'am. Anupam ma'am, have you started recording?
No, I will.
Okay. Ma'am, now I start.
Yes, ma'am. Go ahead, ma'am.
Okay. Respected VC sir, Director sir, Director Campus, Registrar sir, CFO, Dean Mr. Ashtani sir, Department of Engineering and IT, Honorable Keynote Speaker, Dr. all Program Coordinator, all participant, faculty member, non-teaching staff, and dear student. Good afternoon all. On behalf of Department of Engineering and IT, Arkajan University, Jharkhand, I, Dr. Chandraprabha Sahu, welcome you all in this national webinar titled Facile Synthesis of Multifunctional Thiazole-schiff base derivatives via multi-component reaction and their anti-cancer evolution and molecular docking studies. We are very much fortunate for having Dr. Archie Sharma as a speaker for today's webinar. Before we start, I would like to introduce her to all of you. Dr. Archie Sharma has completed her MSc in Organic Chemistry from Guru Ghasidas Central University, Bilaspur, Chhattisgarh. Thereafter, she holds a PhD in Synthetic Organic Chemistry from National Institute of Technology, Raipur, Chhattisgarh. She has experience of more than 7 years in academia and research. She has published more than 9 research papers, including 2 review papers in well-reputed international peer-reviewed journals. Her research work received 5 H-index and 47 citations in a very short span of time. Not only this, she has also co-authored of 2 books, that is Advantage in Structure and activity relationship of coumarin derivatives and dihydroacetic acid and its derivative. Useful synanthons in organic synthesis under the renowned Elsevier publication. Apart from her research work, she is actively involved in teaching UG, PG, and engineering students. Currently, she is working as an assistant professor in Modi Women's University of Science and Technology, Rajasthan. Now, I would like to invite Dr. Archie Sharma to proceed with the presentation, and for others, kindly please mute your mic. In the end, she will be taking few of the questions. Over to you, ma'am.
Okay, ma'am. Thank you so much to Arkajan University, Ashwini sir, Assistant Dean, Department of Engineering and Polytechnic, and Chandraprabha madam for giving me this opportunity. Very good afternoon to all of you. So let us proceed with this webinar. So facile synthesis of multifunctional thiazole-schiff-based derivatives where multi-component reaction and their anti-cancerous evaluation and molecular docking studies. Heterocyclic compounds are the cyclic compounds which contain heteroatom as a part of the ring other than the carbon atoms. Okay, heteroatoms which are other than the carbon atoms, which is present as a part of the ring, is known as heterocyclic compounds. Various amino acids consist made up of these heterocyclic compounds, and we know that the amino acids are the building blocks of proteins. Similarly, some vitamins are also consisted with heterocyclic compounds, and some alkaloids are also made up of these heterocyclic compounds. So in general, we can say that the heterocyclic compounds are very prominent and potent organic compound. Beside these, they are also applicable as solvent, catalyst, cosmetics, pharmaceutical compounds, agrochemical compounds, fluorescent compounds, organic LEDs, and biotechnological compounds. In pharmaceutical industries, heterocyclic compounds are used to synthesize various types of drugs. For example, quinoline is present in anti-malarial drug, then lactam ring is present in penicillin, and imidazoles is present in the metronidazoles. Imidazoles as metronidazoles. Thiazoles. Thia word is used for the sulfur, and azo word is used for nitrogen. So sulfur and nitrogen containing heterocyclic compound is known as thiazole. Thiazoles are having potent bioactivity. So it can shows antimicrobial, anti-inflammatory, antibacterial, and antifungal properties. So why there is need for designing of the drugs?
Excuse me, ma'am.
Yeah, ma'am.
Ma'am, your slide is not moving, ma'am.
It's not visible.
Is visible but is not moving. It is in the first slide. Or
Now it is moving.
yes, ma'am.
Okay.
Yes, ma'am. Ma'am, please continue.
Okay. So amino acids are made up of heterocyclic compounds. Then vitamins and alkaloids are also made up of heterocyclic compounds. Other than that, they are also applicable as solvent, catalyst, cosmetics, pharmaceutical compounds, agrochemical compounds, and fluorescent compounds. Organic LEDs are also made up of heterocyclic compounds. Thiazoles are sulfur and nitrogen containing 5-membered heterocyclic compounds, and it can be used as antimicrobial, antibacterial, antifungal, and anti-inflammatory compounds. The next is need for designing of a new drug. Usually the habitual drugs or habitual medicines does not impart effective role in a particular conditions. That is because they are having the short life span. Then resistivity against drugs. Most of the pathogens, microorganisms can develop resistance against the drugs. Then another shortcomings of the drug is low specificity of the drugs. Some of the drugs like anti-cancerous drugs, then anti-inflammatory drugs, and some neurodegenerative drugs. That means anti-Alzheimer's drugs. They're not very specific and they can cause various types of side effects, severe side effects. Then the poor bioavailability of the drugs. When we intake the drugs, it goes through several biological process like absorption, metabolism, excretion, and distribution. So sometimes these drugs are not available at the target site. Okay. So the poor bioavailability is also a major problem. The next is the cost of the drugs. This is not the least, but it's very important. The high cost of the drug is also very problematic, especially in the developing country like India. So all these drawbacks always encourage us for designing and developing the new drugs. So my objective was to synthesize novel polyheterocyclic compounds and to select the most appropriate synthetic approach for the synthesis of heterocyclic compounds, and then application of nanocatalyst in synthesis of heterocyclic compound, and to investigate the in vitro and in silico anti-cancerous activity. For the optimization reaction, for the optimized and effective reaction condition, I have selected the multi-component reaction. For this reaction, more than 2 starting compounds are required. And the beauty of this reaction is that almost all the atoms of the starting materials can be found in the product. For example, this is the Stecker synthesis. When naphthaldehyde, hydrogen cyanide, and ammonia. These are the 3 starting material. When reacts, it gives the naphthaldehyde derivatives, which contain cyanide and ammonia. That means you can easily see that in end product, almost all the atoms of the starting material is present. In organic chemistry, it is very difficult to synthesize 4-membered heterocyclic compound because of the ring strain. But by using MCR, we can synthesize the beta-lactam ring. Then also by using MCR, we can synthesize anticoagulant drugs. Various types of compounds we can synthesize by using MCR, multi-component reaction. So basically MCRs are useful in synthesis of natural products, medicinal compounds like antimalarial drugs and anti-cancerous drugs, and they are also useful for synthesis of various heterocyclic compounds. Now let us proceed with the synthesis of multifunctional thiazole-schiff based derivatives. Before synthesis of thiazole-Schiff based derivative, we have to synthesize some starting material. Herein, the sulfonine chloride derivative, when react with benzoate, aminobenzoate derivatives, it gives the sulfonamide intermediate. This intermediate further reacts with the methyl pyrimidine derivatives, and it gives the acetyl intermediate, gives the compound which is having the acetyl group. Further, this acetyl group was brominated to obtain the alpha bromoketones. Okay, by this alpha bromoketones are very reactive site, especially for the synthesis of heterocyclic compounds. So to generate this alpha bromoketone, we have to brominate the synthesized acetyl derivatives, okay, by using N-bromosuccinimide. For the confirmation of formation of synthesized compounds, some characterization techniques were used. So this is the H1 NMR spectra of this compound. H1 NMR spectra shows the number of hydrogen present in the compound and also the types of the hydrogen present in the compound. Herein, only one carbon, sp3 hybridized, only one sp3 hybridized carbon is present. So hydrogen substituted with this carbon has shown the singlet peak around 4.939. Then hydrogen which is substituted with this nitrogen, sulfonamide nitrogen, it gives the peaks around, singlet peaks around 8.538. Also the total number of hydrogen is 10, and this is shown in the bottom of the spectrum, total number of hydrogen. Okay. Here also it is 10, and in this compound the total number of hydrogen is 10. Next is C13 spectrum. Carbon-13 spectra gives the idea or gives the confirmation of formation of compound by giving the number of carbon atoms and the types of the carbon atoms. So again, sp3 hybridized carbon atom has shown the peaks around 63.02. Then the carbon which is attached with the 2 nitrogen group and 1 chlorine group has shown the peak around 194.83. Also, the number of the carbon is 17 here. The number of peaks is also, this number of peaks reveals that the 17 number of carbons is present. Then this is the mass spectra of the compound. So the molecular ion peak is 520, whereas the base peak is 520. Then after synthesis of starting material, we have prepared some nanocatalyst, especially the nickel ferride nanocatalyst. For that, we have to take aqueous solution of ferric chloride and nickel fluoride, and we have to add the dropwise sodium hydroxide we have to add. Then it was precipitated and filtered and dried and calcinated. Then white color precipitate was obtained and further considered for the characterization process. So for the characterization of nickel ferride nanoparticle, we have nickel ferride nanoparticles, XRD was performed and HR10 was performed. Diffraction peaks at different HKL plane shows the formation of nickel ferrite, and the formation of nanoparticle was confirmed by the HR10, which shows the size of the average size of the particle is 15 nanometer. So this is the main escape. This is performed by the multi-component reaction where the alpha bromo ketone derivative, the alpha bromo ketone derivative reacts with thiazole-semi-carbazide. When this alpha bromo ketone derivative reacts with thiazole-semi-carbazide, it gives the thiazole derivative. Then this thiazole-semi-carbazide, the amine group of this thiazole-semi-carbazide react with this aldehydes and forms this Schiff's base. So this is the synthesis of thiazole-Schiff's base derivatives. Also, we have taken nickel ferride nanoparticles as a catalyst, and this reaction was performed in ethanol and water. Several reactions were carried out to optimize the reaction conditions. So in initial reaction were carried out without the use of catalyst by using different solvent, and the yield was not very good. It was found that the yield was not very good. Also, the reaction time is of 4 hours. After that, I have taken 5 mg of nickel ferride nanoparticles, and it gives the 89% of yield within 1 hour of the reaction. Then I have taken also zinc ferrite nanoparticles, but it gives poor yield. Then I have increased the amount of nickel ferrite nanoparticle for 5 mg to 10 mg, and it gives the 92% yield. But the increment in the yield from 5 mg by using 5 mg nickel ferrite nanoparticle to 10 mg nickel ferrite nanoparticles is very, very less. Also, then next what I did is I've taken 3 mg of nickel ferrite nanoparticles. Okay. And the yield was only 75%. So I've chosen 5 mg of nickel ferrite nanoparticle as in a standard protocol. So with the 5 mg of nickel ferrite nanoparticles, I have taken different types of solvent, but the result was not very good. So finally, this was the standard protocol selected by me. I have also checked the reusability of the nickel ferrite nanoparticles, and it was shown that the nickel ferrite nanoparticles could be used more than 10 catalytic cycles without losing its catalytic activity. Okay, we can use this nanoparticles, nickel ferrite nanoparticles, for more than 10 times. And by using these nickel ferrite nanoparticles as a catalyst, I have synthesized total different 11 types of derivatives. So again, for the confirmation of synthesis of thiazole-Schiff's base derivatives, H1NMR spectra was, and it shows the peak that methoxy group. Here you can see that the methoxy group is present. The carbon which is attached with this oxygen is actually sp3 hybridized. So this carbon is having 3 hydrogen, which shows the peak around 3.885. Then hydrogen which is directly attached with the oxygen has shown the peak around 10.79, and the hydrogen which is attached with the with this nitrogen has shown the peak around 10.23. Total number of hydrogen in this compound is 18, and this also shows the number of hydrogen, total 18 number of hydrogens. Then this is the C13 NMR spectra of the compound, and it shows that sp3 hybridized carbon atom having the peak singlet peak around 55.10, and the carbon atom which is directly attached to two nitrogen groups and one chlorine atom has shown the peaks around 183.54 nano. 183.54. So this is the mass spectra of the compound, and it shows the base value of 646. All this spectral data confirms the formation of synthesized derivatives. So the next part was to synthesize and to analyze the cytotoxic activity of the compound for different cancer cell lines. 3 cancer cell lines were selected. First is skin malignant melanoma A375, epithelial cervix adenocarcinoma, that is HELA, and human breast carcinoma MCF7 cell lines. Skin malignant melanoma is responsible for the skin cancer. Epithelial cervix adenocarcinoma is responsible for the cervix cancers, and human breast adenocarcinoma is responsible for the breast cancers. All the cell lines were maintained in Delbecque's modified Eagles medium, supplemented with 10% fetal bovine serum and penicillin, with the use of penicillin and streptomycin, or this. The cell viability was assessed by MTT assay, and in vitro half-life inhibitory concentration of all the derivatives against cancerous cell lines were calculated. So this is the in vitro half-life inhibitory concentration of all the derivatives against cancer cell lines. As compared to anatonib, all the derivatives have shown very good IC50 values. The lower the IC50 values, that means they are the higher the potency is in their compounds. We can see that all these compounds are capable to show inhibitory effect for A377 and MCF7. That means they are capable to show inhibitory activity for skin cancer cell lines and breast cancer cell lines. But unfortunately, they are not capable to show any effect on HELA cell line, that is cervix cancer cell lines. So among all the derivatives, these 5 derivatives are capable to show very good inhibitory IC50 values as compared to anatonib as a standard drug. Then a statistical graph is also plotted between the percentage cell viability and the concentration of anatonib, or the concentration of the given compounds. Percentage cell viability is nothing but the percentage of living cancerous cell lines. So as we increase the concentration of the compound, we can see the decrease in the percentage of cell viability, or the decrease in percentage of cancerous cell lines. So as compared to the standard drug, all the derivatives have shown very good anti-cancerous properties. But they are not working, again, they are not working in HELA cell lines, but they are very effective for A375 and MCF7 lines. That means they are very effective for the skin cancer effect, and also for the breast cancer cell lines. So as compared to this, compound 4G and 4H have shown very good percent cell viability, decrease in percent of cell viability. Okay, you can see this. As compared to anatonib, all these compounds have shown very good percentage cell, decrease in percent cell viability. So all these derivatives are bioactive derivatives. Let's say on the basis of all this in vitro analysis, we can say this, these derivatives are the bioactive derivatives. Next is in silico molecular modeling studies. For the in silico study, we have to take, we have taken a 3D structure of HSP90 was generated using homology modeling with the prime application of Scoville. So this HSP is a is the short form of heat shock proteins, and they are usually generated when any cell is in exposure to stress conditions. So if any cell is in contact of any carcinogenic compound, then they are responsible, they can generate the HSP protein inside the cell. So among various types of HSP, we have taken HSP90 for the docking studies. Next is binding site generation. In protein, we know there is very different types of active sites are present. So based on the site score of the software, we can select the best site for the binding of the ligands. Third is ligand preparation. For ligand preparation, ligand prep application was employed. During the ligand preparation, we can remove the water molecules, or we can incorporate the water molecules, or we can remove the hydrogen atoms. These there are different types of parameters in this applications. We can use some of them for the modification of the ligand. Then docking of protein and ligand. Next step was to synthesize. Sorry, next step is to dock the compound with the proteins. So this is the flow chart of the docking studies. First, we have to take 3D protein structure, and then we have to optimize it 3D structure by again we can set different types of environment for the proteins. Then we can create and save energy for the grid maps. Grid map generation of a grid, that means generation of binding site or selection of a particular binding site in a protein. Then optimized with the incorporation of optimized 3D ligand structure, we can dock the compounds and get the scores and docking profile. So all the compounds were docked, and we have taken dabrafenib and erlotinib as a reference drugs. Here, the docking score, as compared to the dabrafenib and erlotinib, all the compounds have shown very good docking scores. Docking score is nothing but the energy released during the formation of bond between the ligands and the protein. And this energy is in the form of kilocalorie per mole. So as compared to the dabrafenib and erlotinib, these derivatives have shown very good docking scores. So on the basis of the best docking score, some of the compounds were selected. for the further docking for profile. So these are the derivatives which were selected for the docking studies. So this is the in silico docking profile. And it shows that as compared to andratolib, allotinib and dabrafenib, all the derivatives have very... So all these derivatives have shown very good in silico binding capacity for the HSP90. All these derivatives, compound 4G and 4H has shown in vitro as well as in silico properties. Very good in vitro and in vitro anti-cancerous properties and in silico anti-cancerous properties. Non-inhibition for all the selected cytochromes enzymes. Then HERE is human erythro-go-go related gene, and this is responsible for the cardiotoxic effect. This parameter is responsible for the cardiotoxic effect. And it should be, the range for this HERG should be below -5. Solvent, but all for anti-cancerous drug compounds. 75, that is skin cancer cells, and MCFs and breast cancer cell lines. The newly synthesized compounds were shown good to an excellent binding affinity for HSP90 proteins. And in ADMA analysis, the best for the absorption and metabolism is shown by the compound 4E, while